Our first speaker this afternoon is Jason McQuirk. Jason has led acoustic telemetry studies that evaluate the effectiveness of engineered structures as deterring juvenile salmons. Recent projects such as Georgiana Slough non-physical barrier study, and that was, I think, mentioned this morning, and the head of Old River Bridge, or Old River evaluation, yeah, the head of Old River Bridge. Sorry, I worked on Old River Bridge too many years. So basically including survival and predation study components. He's actively involved in planning of acoustic telemetry and predation studies on the Sacramento and the San Joaquin rivers. He is a program manager at the Department of Water Resources, and through his leadership at DWR has helped advance research on fish predation. Jason?
All right.
Jason? Thanks.
Thank you.
Thanks.
Welcome, panel members. Again, I am Jacob McQuirk, and I'm here to present on some of the work that we've been doing on salmonid predation. The mic, thank you. Do excuse me, I am a licensed civil engineer, not a biologist, so if you start throwing Latin at me, I'm just going to give you a weird look. All right, so we're going to cover acoustic telemetry. I'm going to cover real quickly the difference between like a salmonid track versus a predatory fish track. We're going to also look at some of the active acoustic imaging we've done as well. That includes mobile hydroacoustics and fixed hydroacoustics, including like Didson and split beam sonar. We're also going to cover a little bit of the predatory sampling we've done, and I'm going to very briefly also cover some of the Clifton Court Forebay predation studies that we have worked on. I am a lot less involved in the Clifton Court stuff, so I'm not going to be able to answer detailed questions. Go right ahead with questions as I present. You can interrupt me, there's no problem, or we can do those at the end. So some of the recent and ongoing work that's being done or has been done is we have done predation studies looking at pre-screen loss within Clifton Court, Forebay. We've done studies at the head of Old River. We installed a bioacoustic fish fence there, which is a non-physical barrier that uses light, sound, and bubbles to try to deter juvenile Chinook salmon, keep them in the San Joaquin River. We did that for 2 years. Recently, in 2012, we installed a physical rock barrier there to also keep the juvenile salmonids in the main stem of the Sacramento. We've also done some acoustic telemetry studies at the temporary agricultural barriers, looking at predator-prey interactions there and passage through the barriers. And some of the work is also coming out of our Georgiana Slough non-physical barrier evaluations, where we looked at a large engineering structure at the divergence of Georgiana Slough from the Sacramento River, and we looked at ways to divert the fish and let the water flow down Georgiana Slough. So some of the researchers, I'll just cover this. We've worked a lot with USGS, Reclamation, various consulting firms, DWR staff, and the Fisheries Foundation. So acoustic telemetry, real quick, what is acoustic telemetry? Acoustic telemetry is a mark recapture study where we implant acoustic transmitter in the fish, and then we use hydrophones to see if I can actually make this work. Hydrophones that will be able to triangulate the position of the fish. A lot of the work that I'm going to show you is based off of an HTI acoustic telemetry system. And one of the benefits of an HTI acoustic telemetry system is it does enable you to get 2D tracks. So like, for example, I could show you real quickly a 2D track. And this is an interesting track at the head of Old River, where you've got originally what we thought was a juvenile salmonid being preyed upon, but later on we confirmed that it was actually a steelhead because we resalvaged it, and it turned out that it was just going with the flow fields. At the head of Old River, we spent a lot more energy actually to reproduce using acoustic Doppler current profilers and actually reproduce the flow field there, so we understand it in a lot more detail. So one of the things is, how do you determine a predation event in an acoustically tagged fish? It can become a real problem if you're trying to figure out, like, how does this fish interact with a barrier that we've constructed or whatnot, and if it becomes consumed by a predatory fish, then, now it's going to be exhibiting different behavior. We started out with a real qualitative approach, which is through experience and whatnot. We've looked at the tracks, and, we've seen that the track starts making an abrupt change. It starts swimming upstream, doing things that smolt wouldn't do, the less directed behavior. We've also moved now into more quantitative approaches, looking at some different mathematical functions on the movement of that fish and probability distributions to determine when and if it's been preyed upon. So this is just a real quick representation of directed movement that we would see indicative of a juvenile Chinook salmon. This here is at the head of Georgiana Slough, where we constructed a barrier. The barrier is not shown there. 2 fish that are going into Georgiana Slough and 2 that are remaining in the San Joaquin River. Here's some predatory fish tracks. As you can see, the predatory fish has a lot more movement. It's kind of all over the place, where you've got some of your more stationary centrarchids that like to occupy the shore, and then the striped bass that like to stay more in the center channel. So as I said, we're trying to move from the more qualitative, where you're looking at the characteristics of the fish behavior and trying to make an assessment, to some more quantitative stuff. Some of the work we've done is we've looked at, these are radar plots, and so we've been able to say, okay, well, how does a different known fish species move? And so like in 360 degrees, that will show you the direction it's moving. And so if you look at like a juvenile Chinook smolt, as you can see, it's got a real directed downstream movement, which would be, actually I'm pointing with my finger, which would be indicative of a plot like that. Whereas as you get to a striped bass, it's moving in all different directions. And so that's one of the things we've done. Working with the USGS, this is work that's coming from Jason Romine of the Columbia River Research Station. He's looked at a mixture model with probability distributions, which this is all of our tag fish at Georgiana Slough, and then he's doing a probability distribution with 2 different, with the levy exponent and the tortuosity. Both are indicative of the movement of the fish, and then trying to see how well does that fish fit in with the probability distribution. Okay. So one of the things we've been able to do is in 2012 with Georgiana Slough, we did a really good job of being able to both qualitatively and quantitatively. As you can see, our results were real close. We may have missed certain fish species, but the qualitative and quantitative did come out very closely. But one of the things I was going to say, which I'll show you in another slide, is that it's not always that clear to be able to tell whether or not a fish has been consumed or not. Sometimes a predatory fish may behave with directed movement like a smolt, and some of the probability distribution work we've done doesn't really look at the direction or the timing per se. It looks more at these specific metrics. And so this is more back on the mixture model. And so what we've got here is we've got, this is, let's see if I can remember what kind of fish this is. This is a predator. So this was, I believe it was a striped bass that came into the area. And so on the first pass, the model determined a probability of a 1.0 that it is actually a predatory fish and not a smolt, even though it started out as a smolt. And then, but by the 4th pass, it had started to, the 4th pass through the array, it had started to exhibit more predatory fish, or rather smolt behavior, directed movement. And this one is opposite. You see the fish started with what looked to be very high probability directed movement out of the river reach, but then on its return, it exhibited more predator-like behavior. And so this is kind of an example of this particular model is not foolproof. It does seem having relatively good results, but you can trick the model as well. So, all right. So one of the things we've been able to do by implementing a multidimensional telemetry study, this particular area we're looking at is the head of Old River, and this particular location is at the head of Old River, where we have, we put a rock barrier here, and we've put non-physical barriers here as well. And one of the things that we noticed is that this particular track, which is going this way, was exhibiting behavior that we'd say is more indicative of a predatory fish, of a predation event. Well, one of the things we were able to do is in 2011 we had a large acoustic telemetry system in the Delta, including a two-dimensional array here. Well, we re-salvaged this fish, which turned out to be, it was a large steelhead. And then so we're able to come back and say, well, based on this known data set, we know that this fish was not preyed upon. In fact, it was a steelhead, and we've kind of learned some of the behavior of the steelhead, which can be quite a bit bigger than the juvenile Chinook, are exhibiting all sorts of different funny behavior. And so, so this is just really, this is partly leading towards us better understanding of what I call known fish. So if we know what that fish is, we know what kind of behavior it exhibits, we're able to then start to build like a cache of its behavior and try to better understand that how it's behaving. Other ways to determine predation events, one of the ways is if a tag gets preyed upon, it'll start exhibiting different behavior. I've got a real quick video that I'll show you here on this. And it's a lot longer, but we're not going to play the whole thing. So if you see, we'll go back there. As you see, coming into the array, we got two juvenile, we got Chinook salmon coming in, and then they become preyed upon here. And so at this point, you've got it exhibiting predatory fish behavior, these two tags, which were originally Chinook. And then eventually, as we move forward, continues to exhibit predatory fish behavior. And then at some point in time, right about here, you see the tag will actually become defecated. One tag will be defecated, and then the fish that's preyed upon, the Chinook, moves out of the system. So I know it's kind of not that suspenseful, but it's only a second. So there it is. See, as you see, and then so there's the defecated tag, and then you'll see that the one that's still in the belly of the fish moves out of the system. So
how long a time period is this video?
This one, I'm not. That was probably the order of the better part of a day. I might have to look at the actual video file to tell you that we recorded on this particular study. We had continuous records of the fish for like 120 days or whatnot. So for the most part, we're able to, if a fish stays within the array, we would be able to track it for the whole life of the tag for the most part.
Can I ask another question about that? Have there been studies to look at gut evacuation rate of the tagged smolts?
Well, if there are, I'm not necessarily familiar with those studies, so I'm not sure. This is just a tag. This slide here is showing us again at the head of Old River. We've broken it up into some different zones, and this is the actual acoustic signal for a defecated tag. It starts to flatline, and these are just the locations for, I believe this is 2012. Yeah, 2012 of where we found defecated tags. And so this is a deep scour hole. Like you would think, there was a lot of tags deposited there. We did, as I mentioned earlier, we have done a lot of work at the temporary agricultural barriers. This is, I believe, our Old River Tracy barrier. And what we're showing here is, I'm going to show you a real quick video that it shows that even though there is a delay in passage across the barriers, every once in a while you do get some passage. And you can see that in this case, there's some specific predator avoidance behavior, which I think is interesting. It's pretty quick, but I can do that. Let's see, we'll start right there. And so you'll see the smolt come in, and then if we slowed it down, you would see that the predator, which is still sitting there as a red dot, starts to actually, it makes a directed move and kind of loops a little bit, and then the smolt leaves the hydrophone array. So, all what we were just talking about was all acoustic telemetry, and a lot of the work that we were showing was the 2D and 3D work that we've done to really evaluate how these engineering structures we put are affecting fish behavior, really trying to get at if we're able to deter the fish and whatnot, and what those efficiencies are. A lot of the work that we've done, in addition to the acoustic telemetry, is active acoustics. One of the big questions we've had is, for example, does our barrier or whatnot concentrate predator densities? Does it change the efficiency of predation? So what we've done to get at that question, I've worked a lot with Dr. Horn from Reclamation, is we've done some acoustic imaging using split beam systems, using Didson systems, and also using in a more limited capacity, multi-beam scanning sonar, this is an image from a very expensive multi-beam scanning device that is looking at our non-physical barrier when it was in action. So you can see, I'm actually putting it on the computer. This image here is our bubble barrier, and with using that scanning sonar. This is actually a Didson image there, but through processing, we're able to get densities and other different metrics from the active acoustic data. So this particular slide is showing us the number of fish in the Head of Old River area. Of course, it does do a good job of giving you density estimates and targets that are a certain size range. What it can't do is you can't differentiate between a species. And so one of the concerns I would have at this location is, knowing the area, we've also got a ton of large carp in the area. And so, of course, a carp is not a predatory fish, so that could be problematic. But what we've been able to do with utilizing both the Didson sonar and the split beam sonar and having basically footage at the same time, you can get some species identification from the Didson sonar, and then you can infer like some different movements. For example, when you're looking at the Didson footage, when we had the 2012 rock barrier in place, you can see very, very distinct behavioral difference between the carp and the striped bass. The carp are kind of just moving real slowly, whereas the striped bass are kind of darting in and out around the carp. But as I said, it doesn't give you a species identification, but it does give you some good information about predator location. And so this is the Didson. As I mentioned earlier, you can get some species identification, but one of the problems with the Didson imagery is it's a very, very narrow beam, and so really you're only sampling a small portion of the water column, and then when you start scanning with it and whatnot, you can introduce complexities. This is an image here with the scanning sonar that I showed you guys earlier. We are going to start to use more of the scanning sonar, hopefully next year, on another predator study we're going to do, but we just got very limited experience with that particular instrument. And then one of the things we've done that's kind of a little more foolproof, that you do have a large striped bass because you pulled it out of the water, is we've worked with the Fisheries Foundation, and we've gone out and we've had them sample for predatory fish. We give them certain quotas, and then they'll acoustically tag a proportion of those fish. And so that does help a lot because, like, for example, this large striped bass probably did get an acoustic tag. It goes back into the system. And for example, this year, we've got a large Vemco array out, so it was tagged with a Vemco tag. And so then it'll be picked up upon every hydrophone that it crosses throughout the Delta. So it gives us more information, kind of like a cache of what type of behavior that it exhibits. And we do plan eventually to use electrofishing. We haven't yet. We're planning to, starting as early as next year. And so this is based off of our predatory fish sampling, and you can see it kind of shows the different composition. This is the yellow striped bass, white channel cat, and largemouth. Spotted are the blue, and these are the locations in which they were captured, and this all took place this year. So, and so it's just giving us a good baseline of what's out there. And using that with some of the acoustic imagery, we're hoping can give us a little more confidence in what we're seeing there. So this year we sampled for 42 days. We captured 383 predatory fish, 30 were acoustically tagged, and 82 were fluoride tagged. And this is kind of our sampling. We got the peak. What is that? I can't tell what month that is. And I'm going to talk just a little bit about Clifton Court. And some of the studies we've done at Clifton Court. For those of you that may not know where, here's Clifton Court Forebay, and this is the Forebay to the State Water Project. Our louvers and our salvage facilities right here, our pumping plants are there. And so as Ron Milligan spoke earlier, we operate these radial gates to take water in on a high tide, and then we'll close them off. Well, in order to be salvaged, they got to make it across this large waterway, and it's got really high predation rates. And so we've looked at some different studies to kind of estimate what's the pre-screen loss and what's the mortality basically just along that dotted path or whatever path it takes to get there.
Welcome to the mic, please.
Okay. All right, so this is just a slide showing some of the studies that have been done in Clifton Court, where, as you can see, for all of our losses are really high. You see nothing in which you don't have a very high loss rate. So it's real common knowledge that it's not the best spot to be if you need to make it to the salvage facility, whereas getting through Clifton Court can be quite a challenge, whereas the Central Valley Project just has a direct intake, so you won't have as high of pre-screen loss to go into the louvers at the Central Valley plant. But this is, and we'll talk a little bit more about the studies that we're proposing in Clifton Court. One of the things that that we are proposing is to put a fishing pier to improve fishing access around the radial gates. Currently, there is a ton of people that they find their way out there, they hike their way out there, they're on the wing wall, they're trying to catch fish, and so it already has a pretty big fisherman presence, and we'd like to be able to increase that presence and make it a little bit safer as well. It's not a very safe situation now. And so, if you lower the densities of predators, you might be able to improve the pre-screen loss. So the current studies that we're working on, and I'm not leading any of these studies or managing them, so I'm not as familiar with them, is we have mark recapture studies using salmonids. We've got predatory fish mark recapture. They're doing creel samples, avian samples, and eventually genetic sampling. And they will be doing some bioenergetics modeling. I know there is currently work going on at Clifton Court right now, and I know we plan to continue our study efforts next year, and I think it's for another 4 or 5 years after that. So some of our future research directions, we are working with National Marine Fisheries Service, and we are going to do a predator manipulation study on the San Joaquin in the same area that I've shown you a bunch of other slides in and around the head of Old River. And the goal with that would be, can we remove predatory fish using netting and e-fishing, and then can we improve smolt survival by doing so? It'll also help us understand, recolonization. If you pull out a bunch of predatory fish, how fast do they come back? And it should also give us a lot better information on just the composition of predatory fish. Then we got the fishing pier, like I just mentioned. And another thing that we plan to do is we plan to continue to support the survival studies that are currently taking place on the San Joaquin. That includes for 2014, we're currently planning a survival study on the Sacramento that'll release. It's not 100% official yet, but it'll release probably around 4,000 acoustically tagged juvenile Chinook salmon in Sacramento, and then we will track their their migration out of the Delta with a finish line of Chips Island, and we'll also instrument all of the divergences, so we'll be able to get reef-specific survival probabilities and root entrainment probabilities. And one thing that I think a couple different speakers mentioned, and I think Ron Milligan mentioned as well, is that year-to-year variability is a very big thing, and so you could, one year you may have a low flow year which exhibits totally different hydrodynamics, totally different survival from a high flow year. And so one of the things that we've been doing is we've been trying to, complete as many of these survival and telemetry studies as possible, because you really can't do a great job of predicting what kind of water year you're going to get. And so, for example, at Georgiana Slough in 2011 and 2012, we got a pretty good variance in flow. We had, in 2011, a real high flow year in which we experienced no reverse flows. And then the following year in 2012, we did experience some reverse flows. And then when we go into 2014, we may get like a low flow year, but that's just one of the intrinsic problems with the survival studies is it's really hard to sample all the different water year types and be able to try to differentiate why are we getting the survival we're getting. Is it something to do with the hydrology, or what factor is causing that? And so we're going to continue to support that. We're also going to continue to support building statistics on known fish, known predatory fish, and like the work we did with a lot of the existing data sets. There's a lot of work that can still be done from the data that we've been collecting on the Sacramento and San Joaquin for the last, I don't know how many years it is now, quite a while. And so that's, I believe, my last slide. Yep. So if anybody has any questions, I'd be happy to do my best to answer them.
I had one I would like to ask you about. You have, using statistical models, distinguish a free-living tagged smolt from one that's in the belly of a predator. But have you examined, so then kind of the history of what the wrong thing to do is if you're a smolt, right? The ones that got eaten did the wrong thing. So is there any effort to compare the tracks of fish that made it through successfully, and if they behave differently than those that were eaten?
Okay, so let me make sure I'm getting the question correctly. Have we looked at the statistics or the behavior of the successful fish? And I would, yes, we have. When it comes to evaluating the barriers, like for example at Georgiana Slough, we the first 2 years we tagged around 1,500 fish, and every tag was looked at, every bit of that track. And so what we did first out in 2011 is we had what we called our Fish Fates Conference, and everybody got, and we basically looked at these video files, some of which you saw, of these tracks, and came up with a rule set and tried to determine, was the fish preyed upon, did it go into Georgiana Slough, or did it stay in the Sacramento River? And one of the things we've tried to move forward with is some more quantitative analysis. We've done a lot of different things, and I've kind of just given you a tidbit on some things that have worked. And so, of course, as you all know, there's other things that may not work as well, and some pretty advanced modeling and whatnot that we're still working on. Another research direction that we are taking is we're starting to work with the U.S. Army Corps of Engineers. And this is really specific. A lot of the work I do is specific to how fish are interacting with engineered structures, barriers and whatnot. We're working with Andy Goodwin and the U.S. Army Corps of Engineers to do individual-based fish behavior modeling, where we'll use a computational fluid dynamic model at the actual divergence near our structure. We'll come in with our fish tracks, and then we'll be able to make a predictive model that looks at the multidimensional movement of the fish. And so that's kind of one of the directions we're trying to take with how we design engineering structures.
How well do you think that your test subjects are representing the target species or race of interest? So in particular, looking at hatchery versus wild, the size of the fish that you tag, whether you know what race of fish it is, the tag effects, those kinds of issues. So how well do you think that those fish that you're tagging represent basically the untagged group of the fish that you're interested in?
I I can speak a little bit to that question. Of course, some of our primary investigators would be a lot more knowledgeable on that subject, but I think one of the cold hard facts is we're able to use the fish we can get, and so if you can get hatchery Chinook salmon from Coleman National Fish Hatchery, that's where like all the fish from our Sacramento River studies have come from. There is some information that say they might be larger than some of the native fish, but I think where we're at is we've got to use the fish that we can actually get. We're not in a position where we're out tagging wild fish, and so one of the things that we have moving forward on our Georgiana Slough studies, we are trying to release the fish further upstream, and there's for multiple benefits there. One of the reasons is to try to make sure that the fish is more acclimated and is acting more like a natural, or rather a native fish. One of the things that we'd like to get at, whether is like diurnal holding and moving, is it moving? Is the fish moving more in the evening and night? And so we think that having the fish in the system longer is going to hopefully, there's two things it's going to do. It's going to likely get preyed upon, so we'll have to increase our sample size. But the other thing is we're hopeful that it'll be behaving more naturally.
I guess what I'm wondering is not so much that the challenge associated with getting your hands on natural origin fish, it's how well do you think that the fish that you do have access to do represent the natural origin fish?
Well, on the Chinook side, I mean, the steelhead I think are a little more confusing to me. But on the Chinook salmon side, we do feel that at least with looking at the interactions of those fish with our structures, we do feel like they are acting as if as we would expect native fish to act. And so one of the bigger questions has been on the survival side. So do we need to do more things to try to make them, the survivability be similar? So that's kind of where we're at. But from the research that we've done and from the folks that I've worked with, we do feel that at least how they're behaving with our structures is representative of what we get for native fish. But then again, I think if you go back to an earlier presentation, I think on fall run, aren't we dealing mostly with hatchery fish as well? I think that's the vast majority of the fish on the fall run side.
I have a question about steelhead, or I guess it's specific to steelhead, but it might be applicable in general to the Clifton Forebay. A fair number of some of the studies demonstrated pretty extensive residence times. I think some of the tags, 60 days, tags were still active and perceived to be steelhead, but remaining in the Forebay. And I didn't notice that behavior was part of your focus, because it seems like something might be influencing the rate of movement or precluding that evacuation of the system. I know there's some relationship with export, but are there any directed studies really looking at?
Are you referring to behavior of steelhead within Clifton Court Forebay, or just behavior of steelhead in general?
In this case, more behavior of fish in the Forebay, sort of extended residence times that would obviously increase their probability of predation.
Well, I know I know that our investigators, one of which is Kevin Clark, have looked at residence time, but I do not believe we have put out a two-dimensional array per se in Clifton Court Forebay, and I'm not sure if we plan to do so in the future. One of the issues with such a large Forebay is it would be very equipment intensive and also data processing intensive to be able to track those fish in multi-dimensions, because it's just such a big area.
So the effect of the predation, your focus is mostly on the predators and the managing from that end rather than maybe just tackling the behavior of the fish, or if there are any things that can be done to alter their movement through the bay. I mean, that's not what, so you're not working on that question.
I specifically am not working on that. And so your question is whether or not we're trying to study the actual behavior of the steelhead? And
right, given some pretty extensive residence times in that system, which for a migrating steelhead.
Yeah, so like I said, I'm not the investigator, nor am I leading the investigators on the Clifton Court Forebay work, so I can't really answer that. But what I can say is what we have done on steelhead is we have been able to, I had a slide earlier on there with the confirmed tracks, and so we were able to look at the multidimensional movement of that steelhead in our two-dimensional array at the head of Old River, and it exhibited behaviors that we would have characterized as potentially predatory fish, but then that same fish was then salvaged, and so we were able to confirm that it was not preyed upon. In fact, it was an 11-inch steelhead that was salvaged and then released. So that's one of the things we're able to do to try to get a better understanding of the movement of fish and how it interacts. Because one of the things that we've learned is that the steelhead are a little bit more, they act they're not as predictable in their movements.
So I have two quick questions. One, catfish are predatory fish, but at least in reviewing the literature and listening to your presentation, there seems to be little effort to figure out what catfish are doing with respect to salmonid predation.
We have actually tagged a number and captured a number of catfish in and around the head of Old River. So we do have channel cats, and channel cats are, we do feel, are one of the predators that are consuming juvenile Chinook. So we do have some information on catfish that we collected in and around the head of Old River.
The second question is the Forebay just a death trap for fish? I mean, when a salmonid enters, do they ever escape?
Well, I mean, I think if you're maybe a few % of them might escape. I think understanding just the dynamics of the Forebay is important, and I can speak to that for 1 minute here. And that is, as you come through those radial gates, the velocities are very, very extreme. You've got a deep scour hole just on the other side of those gates, so that'd be the first point in which they may get picked off by predatory fish. They come through, they likely could be tumbling through those gates. First thing they do is then there's probably likely a hungry striper waiting for them. And so if they make it through that, then they've got to make it all the way across the open water, all the way to the area that leads into our louvers and our salvage facility. So I don't know how to answer that.
It sounds like you're saying yes.
Yeah, essentially it doesn't look like a very good neighborhood for fish that enter it. Unless, of course, you're a striper, then it's a good place to feed. All right. That's all I got. Thank you.